studying the unit algae. So this group of organisms are different from algae because according to the Whitaker's classification, which we have studied in the previous class, fungi belongs to a separate kingdom called as mycota. But whereas algae are containing chlorophyll and hence they are photosynthetic in function. Means they are capable of synthesizing their food material. So algae are photosynthetic, so they are capable of synthesizing their food material. But fungi are somewhat different. They are quite opposite in case of nutrition. So these are basically achlorophyllous organisms. So that means they are not capable of synthesizing their food material. So they depend on decaying matter, or sometimes they are parasitic in nature. So fungi are achlorophyllous organisms, and these achlorophyllous organisms, in this we must study about the general characteristics of this fungi. Basically, what are the characters of fungi, and we are studying about the habitat of fungi, and we are studying about the range of thallus organization, means what type of thallus we find in this group of organisms. And also we are studying the cell composition, then we study about the reproduction. So fungi, they are found in all types of environment. They are found in soil. There are certain fungi which are found in water, and there are also saprophytes. So the already I said that they are achlorophyllous organisms. So hence they do not contain chlorophyll. They are heterotrophic in nutrition. They are either saprophytic or they are either parasitic in nature. So when they are saprophytic, they depend on the decaying matter, and when they are parasitic, so they are either endoparasites or ectoparasites. And even they form a kind of symbiotic association with the other organisms. That is also another nature of this. And if you come to the thallus, we've come across a kind of amoeboid type of plasmodal amoeboid thallus, or sometimes there are certain unicellular examples, and there are even multicellular which are filamentous in the nature. That is the type of the thallus we find in this. And then when we come to the cell wall is made up of usually a complex chemical called as chitin, or it is also called as a fungal cellulose. That is the nature of the cell wall in this fungi. Then this thallus, it is or the cells, in other words, when we come across the cells found in the thallus, there may be uninucleate cells, there may be binucleate, or there may be multinucleate cells also. So hence this nuclei which are present, especially in case of binucleate and multinucleate, the nuclei may be not of same type. There may be different sized nature of nuclei. So that is why they are heterokaryotic in nature. That is the type of character. And the nutrition already I said they are either saprophytic or parasitic. So that means heterotrophic type of nutrition is seen. Then if we come to the reproduction, both the types of reproduction is common, sexual and asexual, and even vegetative type of reproduction is also seen in some fungi. That is about just the basic general characteristics of fungi. Then habitat of fungi, or in other words, the ecology. In what type of environment fungi are found in diverse habitats. So different types of habitats we find. So that means most common they are found in the environment or in a medium containing organic material. And there are certain fungi which are found on land. They are terrestrial, where there is humus soil, which is also rich in organic content. So we find fungi growing on land. We find growing in decaying matter, and also in case of parasites, the fungi live inside the tissues of plants and also animals. So fungi are parasitic in the tissues of plants and animals, and there are certain fungi which are aquatic, and such fungi are usually primitive in nature. For example, chytrids, they do not have a complex nature of thallus, so these are found in water. And there are certain fungi which are found on foodstuffs, on certain food items also they are found, and that is the habitat of fungi, which is nothing but the ecology of fungi. Then the range of thallus organization. So what type of thallus we find? So in the general characteristics, just we mentioned about there are certain fungi. For example, this Plasmodiophora fungus. It is having plasmodal amoeboid nature of body, which is having free cytoplasm, and there is no any covering for that. So that means the protoplasm is said to be naked, and it is of course multinucleate. Several nuclei are distributed in this amoeboid mass of protoplasm, and it is usually referred as plasmodium. And then in case of chytrids, which are the lower fungi, they are called as, there the cells are spherical and single cell structure, which are very primitive in structure. And even yeasts are also unicellular. So yeasts or saccharomyces, they are called as. They are unicellular, but most of the fungi, they are made up of some fine thread-like structures which are branched, and they form a cottony mass, and these branched filaments are called as hyphae. So these hyphae are interwoven, and they form a mass which is called as mycelium. So mycelium is present there in the substratum where they grow, and this hyphae which are present or which constitute the body of the fungal body, we can in some fungi the hyphae sometimes they grow over the surface of the substratum, or they grow inside the substratum. And in addition to that, there are certain fungi, certain hyphae in that fungus which grow vertically into the air. So that hyphae which grows into the air, they form the reproductive structures. But the hyphae which is present over the surface and into the substratum, that constitutes the vegetative mycelium. So we can differentiate two types of hyphae or mycelium in such kind of fungus body. Hyphae which is growing over the surface and or into the substratum, and hyphae which are growing into the air vertically, and they produce the reproductive bodies. So such kind of fungi, they are called as eukarpic fungi. So that means they are having two types of hyphae. So what are eukarpic fungi means? They are also containing hyphae, But we can differentiate two types of hyphae: the vegetative hyphae and as well as the this reproductive hyphae, or which form the reproductive bodies into that go into the egg. That is eukaryotic hyphae. Then the mycelium or hyphae may be septate or aseptate. So inside the mycelium or the hyphae, the cells may be having cross walls or may not be having the cross walls. Now, for example, these fungi, when we come to the classification, there are actually four groups of fungi: phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes. So the phycomycetes, they are the primitive fungi actually. They resemble the algae. That is why they are called as algal fungi. So these algal fungi, phycomycetes, they are what are having, they do not have the what are called the septa. Now suppose this is the hyphae, which are the thread-like structures. So these hyphae of the phycomycetes, they don't have the cross walls. Suppose these are the cross walls. So these cross walls are completely absent in the case of the phycomycetes. The nuclei which are present, they are freely present in the hyphae or the mycelium. So when we observe such kind of hyphae, they appear to be multinucleate in nature. And actually they are not multinucleate because it is not a single cell. So such kind of occurrence of numerous nuclei, it is actually called as the stenocytic condition or stenocytic. The hyphae are stenocytic means the nuclei are freely present. That appears to be multinucleate because of the absence of crosswalks. So this is called as aseptate hyphae. That means the septa are absent. So the cytoplasm and the nucleus are freely present in the hyphae, and it is called as stenocytic condition. In the other classes, except the phycomycetes, that is in the other classes means like ascomycetes, the basidiomycetes, the deuteromycetes. So there the hyphae is septate. So the hyphae is septate. Suppose these are the cells. So these are the septum which are present in the hyphae, and we find the nucleus in each cell like this. And though the hyphae are septate, so in the higher fungi, which are actually called the true fungi, ascomycetes, basidiomycetes, and the deuteromycetes, which are septate, they divide the cells, hyphae into cells, and the septum, though which is present there, it is having a small pore in the center. So that means the partition is not complete between the cells. There is no complete partition. So this partition, which we see in the hyphae, so that partition is incomplete, and each septum has a central pore. So there is a small pore in the center, and that means they are not completely separated. This indicates so that the cytoplasm of the adjacent cell and even the nucleus of the cell will pass through the pore across the septum and enter the adjacent cell. And that is why sometimes the cells of the septate fungi or the septate hyphae, they appear binucleated condition, so which is called as dikaryotic. So presence of two nuclei in a cell, that is dikaryotic condition. So the difference between the absence of septa, the presence of septa is actually, though the septa are not completely formed, but however the presence of septa will provide a kind of mechanical support to the hyphae. So this septum wall, which is incomplete, having a central pore, provides mechanical support to the hyphae, and so that is about the range of the thallus organization. So when hyphae are present in the higher forms, means like except yeast and certain chytids which are unicellular, the hyphae may be separate or separate. But when they are unicellular, there is no question of hyphae. A single cell itself is the cell which conducts all the activities in it. That is in case of yeast cells we come across. Then when we come to the cell wall composition, now the cell wall is different from the cell wall of algae. In case of algae, in most of the examples, we have come across that the cell wall is made up of pectin and the cellulose. So the outer layer is pectin, the inner layer is cellulose. But however, in some cells, maybe in Watcheria, there is an additional layer of chitin which forms on the outer surface. So we come across three layers in the cell wall. But here, the cell wall is also made up of chitin, which is also called as fungal cellulose. But this chitin is different of the chitin of animals. So in phylum Arthropoda, especially insects, the exoskeleton is also chitin, which forms the exoskeleton of the body of the insects, like the cockroach and other examples. But this chitin is different from the chitin of animals, and hence it is called as the fungal cellulose or the fungus cellulose, which is not identical to the chitin of the animals, and it is chemically different. And this chitin, which is present in the cell wall and also in the septum, it is present in the form of elongated, and variously it is organized in the form of microfibrils, and they are present in the different layers. They are oriented, and in case of yeast, this chitin is absent. See, they are unicellular, so chitin is common in the hyphae. In the cell wall of hyphae, when it may be aseptate or septate. But in unicellular forms like yeast, chitin is absent. So when chitin is absent, there are different chemicals which are present in the this cell wall. So in yeast, we come across a complex polymer which is called as mannol and B-glucan or beta-glucan. So mannol is a polymer of mannose, which is an hexose sugar, and glucan is a polysaccharide of glucose. That means it is a polymer of glucose. So such cell wall composition is present in the case of this yeast cells, where chitin is absent. So that is the cell wall composition in case of fungi. Now, if we come to the nutrition in fungi, how the nutrition takes place. So basically, in the general characters we mentioned, as they are achlorophyllous organisms, so that is why obligatorily or compulsorily they depend on an external source. It may be saprophytic or it may be parasitic. So here the fungi utilize the organic materials or the inorganic materials from the source of their nutrient supply. So from where they get the nutrient, so from there, from that source, they obtain the organic and inorganic compounds. And these, it may be in parasites and as well as saprophytes. And the basic nutritional needs of a fungi is actually a suitable compound which is a source of carbon and energy is necessary. So a compound which provides carbon and also energy is necessary for nutrition, a basic nutritional need for fungi. And also, in addition to that, a nitrogen source is also necessary for the building up of the cells. So actually, this is also true for other organisms. But in the same way, fungi require the compounds which are the source of carbon for energy, and also a source of nitrogen is necessary. In addition to that, there are certain inorganic materials like sulfur, potassium, phosphorus, and also magnesium, which are required in significant amounts in activities. And the inorganic materials like iron, zinc, they also are required in minute quantities, like magnesium and zinc are also necessary in the minute quantities. In addition to that, certain amount of vitamins and other minerals are also required for their growth. Now, other than nutritional, in case of nutrition in fungi, so merely the nutrition source is not over. In addition to that, the fungi also require some factors which are influence for their growth. So these environmental factors, besides the nutritional requirements that are required, for example, temperature. See, though the all type of nutrients are available, so if the temperature is not favorable for the growth of fungi, so that is why the supply of the nutrition or the nutrients is not proper. So that is why temperature and presence of oxygen supply in that environment, and even the moisture, the pH value, all these are important for the growth of the fungi. That is why supporting to the nutrition, environmental factors also help in the growth of the fungi. Now this fungi, when they are growing in a particular substratum, the hyphae, which I mentioned in case of eukaryotic fungi, certain hyphae grow over the surface of the substratum and also into the substratum, and also there are that is called the vegetative mycelium, and those which grow above into the air or vertical, they are the reproductive bodies or reproductive hyphae, so they are called as eukaryotic fungi. But when the fungi is growing over the surface, it absorbs the nutrients directly from the substratum. So the complete hyphae, the complete mycelium is capable of absorbing the nutrients from the substrate. But in some fungi, these hyphae or the mycelium, they produce some globose or somewhat finger-like structures, which are called as haustoria. So these haustoria are they are some kind of absorptive in function. Sometimes they are lobed or branched into the substratum, and then they absorb the food material. Especially in case of parasites, we come across haustoria. Usually, the saprophytic fungi, when the hyphae is growing in a particular organic matter or substratum, the complete hyphae is capable of absorption of the nutrients. But in case of parasitic fungi, there is a particular hyphae which are called as haustoria. The hyphae produces some kind of globose or somewhat finger-like or lobed branched hyphae, which helps for absorption. They are called as haustoria. And these haustoria, or sometimes the hyphae, in case of parasitic fungi, they enter into the host, and when they enter into the tissues, they may be intercellular or intracellular. The hyphae may enter in between the cells, that is intercellular. Sometimes the hyphae may also penetrate through the cell wall of the host cells and enter into the cell inside the cell, which is intracellular. So that is why hyphae may be intracellular or intercellular, which helps in absorptive in function. Then when we come to the other types of fungi in nutrition, there are also symbiotic fungi. So this is saprophytic and parasitic. There are also symbiotic fungi which are having close association with usually other organisms. For example, in lichens we have studied. Lichens, they are a group of organisms where algae and fungi are in the symbiotic association. So the fungi provide materials to the algae which conduct photosynthesis and provide food to the fungi. There it is a kind of symbiotic association. And this symbiotic association is also seen in mycorrhiza, where certain fungi they are found in association with the roots of some plants, especially in the gymnosperms, phylus. There are certain fungi, for example, a fungi which is called as glomus, and that fungi is present on the roots, and it will have a kind of symbiotic association with the roots of that plant. So it helps the absorption of minerals from the soil, and also the plant provides the food and shelter to the fungus. So that is a kind of mutual help between the two, or a kind of symbiotic association is seen in such kind of fungi. So that is about the nutrition in fungi. Now if you come to the reproduction in fungi, what are the methods which are found in the fungi, methods of reproduction? So reproduction is actually by three ways: vegetative, asexual and sexual. So in the vegetative methods, which are seen in most of the fungi. So in vegetative type of reproduction, so one is commonly the fragmentation, which is common even in algae. Also we come across such kind of reproduction. And also there is a kind of which is called as chlamydospore formation. Chlamydospores which are formed. That is also another type of the reproduction. Then there are structures which are called as oidea, which are formed. They also help in this reproduction. Then there is a kind of what is called as the fission, which is also a type of reproduction. Then there is a kind of method called as budding in some examples we come across. Then there is a kind of reproduction which is called as rhizomorphs. Then there are structures which are called as stenocia, which are skin. So these are some vegetative methods of reproduction. So just to understand what are these, in case of fragmentation, already we have mentioned that the hyphae which are present in the body of the fungus, when they get accidentally separated into fragments, so each fragment is capable of giving rise to a complete body of the fungus. So instead the fragment or the separated segment does not die, or it does not get damaged, so that becomes a separate new body of fungus by fragmentation. Then chlamydospores, these are also spores which are formed, which are just like endospores as in bacteria, and even in the nostoc or chlamydomonocycle, there also these endospores, they do not, they form a thick cell wall, and they have having a particular amount of reserved food material, and these can resist desiccation, dry conditions or desert conditions, and thus that they can overcome unfavorable conditions. So in the forthcoming favorable conditions, the cells the cells get removed from the thick wall and then start dividing and give rise to further development. That is in the case of endospores. So it is almost similar like the formation of endospores in some organisms. Then if you come to the next another method, in some fungi, we see that oidium is present, which are also called as arthrospores. So oidia or arthrospores, it is also a structure which gets separated from the hyphae, and each separated segment, it is somewhat round in shape, but actually they are not spores like chlamydospores, but these are separated structures which are somewhat spherical in shape, and they get separated. So when they get separated and they reach or fall on a suitable medium, so they germinate and give rise to a new mycelium. That is how the oidia is best. Then fission, it is a simple cell division. For example, in case of yeast, we come across this. So it is the yeast which are unicellular. They simply divide. And even in the case of hyphae also, the cells divide by simple division then. So just the nucleus divides into two, and a cell wall is formed in the center, and thus two cells are formed, and those the daughter cells, they acquire the size of the parent cell. Then in case of budding, so that is the fission is simple division. But in case of budding, what happens? The cell, it produces a projection at one end, like this. Suppose this is the yeast cell. So at one end of the cell, a bulb-like structure, a, a bulging of the cell wall or a swollen structure appears, and the nucleus divides, and one of the nuclei enters into the extended part or the bulged part, outgrowth, what we can call it as. So this outgrowth enlarges further and thus separates from the parent cell. So like that, several buds may appear. So each outgrowth, each bulge portion that originates from the outer part of the cell wall or outer part of the cell or on the surface of the cell, it is called as bud, and that process we call it as budding. So this budding gives rise to a chain of cells, and each new cell which is formed, it further gives rise to another bud, and like that a chain of cells may be seen in such kind of fungi, especially in yeast. Actually, yeast is unicellular. We find each cell is independent, but sometimes when the cells are found in a row or a filament, it is appearing like a mycelium, septate mycelium, hence it is called as pseudomycelium, but it is not true mycelium. But however, the cells will later separate and live independently. But in the rhizome, what happens? There the hyphae, which are usually present in the substratum, they form strands and appear root-like structures. So these hyphae, they form cord-like strands, and these strands, they are the means of the vegetative method. So they get separated. So the hyphae, they actually form cord-like structures, which are a group of strands there, and that structure is called as rhizomorphs. Rhizomorph means a root-like structure, a structure which resembles a root. It is not a true root, but it appears like a root. It separates from the main other hyphae, and thus can go into an independent fungus or independent organism. Then sclerotia are also another group of structures. So these sclerotia, there are some soft structures. Sometimes they are cylindrical in shape, or sometimes they are somewhat irregular in shape. So these constitute a dense mass of hyphae, which is somewhat thick walled than a normal. And this thick walled hyphae, which is having some short segments, so that structure is called as sclerotium, and thus it will separate from the main hyphae, and thus it will divide into a new organism, or it produces cycle. Then if you come to the asexual type of reproduction and sexual type, what are the methods which we come across in the asexual type of reproduction? In asexual type of reproduction, we come across two types of spores which are produced. Spores which are produced by meiosis and spores which are produced by mitosis. So they are referred as meiospores and mitospores. So meiospores and mitospores, they are haploid and diploid. Now in case of these spores by meiosis, so as the spores are haploid, but in the case of mitospores, they are diploid. And in mitospores, we come across two types of spores. They are called as sporangiospores, and there are conidia or conidiospores. They are called. They are produced in a structure called as conidium. So sporangiospores and the conidia, they are present in the certain fungi, and especially in the aquatic fungi. So there, the aquatic fungi, they produce the sporangiospores, and these sporangiospores are motile, and they are usually called as zoospores. And then the non-motile, that means we come across two types of spores there, motile and non-motile. So the motile sporangiospores are called as the zoospores, and the non-motile are called as the aplanospores. And these aplanospores, they are characteristic of the terrestrial species. Fungi which are present in the terrestrial, they are aplanospores, and the conidia are also non-motile, which are produced by certain fungi. And these conidia, sometimes they are directly produced from the mycelium, or sometimes they are produced in a structures called as conidiophores, which are specialized structures of the fungi which produce the conidia. That is how the asexual reproduction takes place. Now, if you come to the sexual type of reproduction, so the sexual type of reproduction, it involves three types: isogamous, anisogamous, and oogamous. So in algae we have studied this. The same thing here will come across in the fungi. Isogamous means the gametes are similar. Anisogamous means the gametes are not similar. Oogamous means there are specialized organs there. So these two are called as this heterogametic in structure. So that means when the three types of reproduction when we come across, so this isogamous and oogamous are heterogamous. That means the gametes are clearly differentiated. The male and female gametes are morphologically identified. So in isogamous, what happens? Same type of gametes. Now whether it is isogamous, aniso, or oogamous, but we come across three stages in this, like plasmogamy, then karyogamy, and meiosis. So plasmogamy, then karyogamy, and meiosis is the three stages which we come across in this. So in plasmogamy, what happens? When the two cells fuse, the plasma, that means the cytoplasm, the protoplasm will fuse each other. That is plasmogamy. And after the fusion of the two cells, the male and female nuclei, when they fuse, we call it as karyogamy. Sometimes they may not fuse; they remain separately, which we call it as dikaryotic condition. the nuclei will separate, and that is called as dikaryotic condition. Then after the two nuclei which are present there, they later may fuse. There is the dikaryon, which we call it as that is condition. The dikaryon may fuse, and then it will form a diploid nucleus, and then it is called as synkaryon. The two nuclei will fuse and form a fused diploid nucleus. Then the diploid nucleus it will undergo meiosis and produce the meiospores. Then it will produce the meiospores. That is in case of this. Then in case of isogamous and oogamous, already mentioned, the gametes are clearly differentiated. They differ usually in their size. The male are smaller and female are larger, and the male are motile and usually the female are non-motile. So based on that morphological differentiation, we can see which is male and which is the female. That is how the oogamic type of reproduction takes place in this fungi. Then after this sexual type of reproduction, when we come to the classification, so the simple classification is there. So the fungi are divided into four divisions. That is four classes we can mention: class Phycomycetes, class Ascomycetes, Basidiomycetes, Deuteromycetes. So Phycomycetes are algal fungi. Ascomycetes are the Ascomycetes are sap fungi, and Basidiomycetes are club fungi. Deuteromycetes are imperfect fungi. So then the Phycomycetes, they include several examples like the Phytophthora, albugo, and so on. Ascomycetes includes yeast like Saccharomyces, Penicillium, and many others. Basidiomycetes includes Agaricus, and Deuteromycetes includes the Alternaria and Trichoderma, and so on. So that is about the classification of the fungi. Then before going to the study of this types of the fungi, so there are certain economic importance of this algae which are related to the algae. So in the fungi also we are studying certain economic importance. But before going to the types like Rhizopus, Penicillium, and Agaricus, then Paccinia, and many others. So this economic importance of algae when we go through. So the algae are significant to the ecology. Particularly when we see the distribution of the algae, nearly 50% of the photosynthesis which takes place in the earth or in the world, it is contributed by algae. So that means this shows that about nearly 50% of the oxygen which is released into the atmosphere is by algae. So though they are very small, but the population and their distribution is very vast, and that is why they contribute a large amount of photosynthesis to the atmosphere or in the world. That is the significant contribution of algae. And algae are also fish to the aquatic organisms, food to the fish, especially and other aquatic organisms, especially green algae. It is a source of food for the fish and also other aquatic animals. That is the another important of algae. And there are certain algae which produce antibiotics. For example, chlorella is the name of a green algae which produces a antibiotic called as chlorella, and this chlorella is a antibiotic that is one of the significant uses. And the blue-green algae, which we have already studied, like Nostoc, Oscillatoria, so those algae, they contribute fertility to the soil by nitrogen fixation. as they are commonly found in the soil moisture. So they fix the atmospheric nitrogen and bring about nitrogen fixation and increase the fertility of the soil. And certain blue green algae, which are especially surrounded by mucilage sheaths, they help to control the soil erosion because they bring about aggregation of the soil particles. And when they bring about the aggregation of the soil particles, so the loose unbound particles, so they are aggregated properly and they prevent soil erosion, and thus they improve the physical texture of the soil. That is the another significance of the blue green algae. And the diatomaceous earth, the diatoms, which after death of the cells, so they give rise to, or the cell wall after death. So it separates. It consists of pectin, silica, and all those. So that cell wall, which is made up of silica and pectin, it gets deposited, and thus the cell wall will form gradually over many years to form the diatomaceous earth. So this diatomaceous earth is used, it is removed from the floor of the ocean, and then it is used for various purposes. It is used as a filtering agent in certain oil industries, and also it is used as an insulation material in case of furnaces, and also it is used as a soundproof material in the construction of walls, and also it is used for the manufacture of metal polishes, and also it is used as a to improve the resolving power in case of lens, especially in microscopes and other cases where the lenses are used. So that is the uses of the diatomaceous earth, and for various purposes these are used. And most of the brown algae, including Sargassum, Laminaria, Fucus, and any others, so they contain, or they are rich in minerals, carbohydrates, and certain vitamins, which are used as food in certain countries. For example, Laminaria is used, and ulva, which is commonly called as a sea lettuce, it is used as a food in certain countries, particularly in Japan and some countries. And this laminaria is also used to produce a kind of standard food called as kombu, which is standard food in Japan and commonly used by the people there. And that is an important product which is produced from laminaria. And there is a algae which is called as Nereocystis. So the air bladders and the types of this algae, they are used in making a kind of food product called as citron. So this citron is a product which is produced from that algae, and it is used as food. And certain chemicals like alginic acid, carrageenan, agar is also obtained from algae. So this alginic acid, which is obtained from brown algae, especially like laminaria and fucus. So that alginic acid is used for various purposes. For example, in stabilizing emulsions, in the manufacture of flame-proof plastics, and also in security glass and certain artificial fibers. There it is used, and thus that is the use of arginic acid. So carrageenan, which is obtained from some red algae, so that is used as a cleaning agent in case of textiles and leather and weaving industries. That is another product which is obtained from red algae. And similarly, the agar, which is another important product from this brown algae like Gelidium and Glyceria, that agar is a product obtained. It is used in as a component of culture medium. So whenever microorganisms, microbes like bacteria, they are to be cultured in the lab. So their agar is used in the culture medium, which is an important component, and it is also used in the canning of fish and preparation of ice creams and as well as certain jellies. So like that, algae are also having several economic importance which are used for various purposes. So that is about the introduction of fungi, and in the next part we have to study about the rhizopus plant history.